Hi everyone, I'm Justin, and today I'm going to talk about bioinformatics. Let's get into the topic. Why I choose that topic? Because I find it interesting and challenging. Also, completing the DNA sequence can also make us have a bigger understanding towards the genomic data of each organism. The word bioinformatics means an application of combining biology to computer for biological analysis. For example, in criminal scenes, we can use a hair or fingerprint of a person to determine its DNA sequence in order to figure out whether the person is a culprit or not. Why people want to do it? The answer is very simple. Because they want to know more about biology. And they want to have more desired traits and get rid of certain pathogens. Next, this is the main flowing chart of bioinformatics. First of all, on the left, people should collect their target DNA in order to map out the whole genome structure. Then the DNA will be cut into segments. Next, take out the specific segments inside these segments. Then library, which brings a place for database storage, starts to select the DNA. Later, among these leftover segments, some parts are selected for amplification, which means replication. These DNA segments will be sent to different analysis stations such as Roche 454, Illumina, BLAST, or FASTA, etc. Next, these machines analyze and put these genes together and can see the function and sequence of these genes. This is the overall summary of my explanation of the process of bioinformatics. In this process, many specific machines are required. I will explain some of them. Roche 454 is a biotechnological company that uses GXFLX system to predict the shape of the protein by looking at the magnified sequence taken from the organism. And Illumina is another website for SCS machine analysis that helps sequencing the DNA and regulate the gene. BLAST is used to align and identify proteins. The picture shows the aligned genes of different animals, which they have the similar amount of bases. Bioinformatics can use this to identify common bases in specific areas, which is marked by different colors. By looking at this graph, we can know that monkey and human have extremely similar gene and far-related organisms have less similar gene alignment. And FASTA, as similar as BLAST, is used to compare different DNA sequences to find out the common similarities and differences of DNA sequences between different organisms. As you can see in the picture, different sequences are put together for comparison. So FASTA is used to compare different segments of genes. Next, I'm going to talk about its advantages and disadvantages. In advantages, people can have further understandings in the field of genetic engineering. Secondly, in criminal scenes, people can catch the culprit easily by DNA scanning through the process of bioinformatics. and we can prevent organisms from dying of certain diseases. Also, gene cloning, or not gene cloning, is polymerase chain reaction, require the knowledge of the DNA in order to have desired traits. Then I'm going to talk about the disadvantages of bioinformatics. First, some people might feel a sense of insecurity because their DNA is screened, because every genetic info in their bodies is shown to strangers. Secondly, it might take a lot of time for searching databases. Sometimes it might take a lot of days to search for a whole human genome structure. Okay, here's my conclusion and connections. All in all, after searching bioinformatics, I realized that there are a lot of things and other research topics that are closely related to bioinformatics. For example, in polymerase chain reaction, we can have a billion of identical segments by replicating from one segment of DNA. I will explain the process of polymerase chain reaction. So first of all, separate a double-stranded DNA by adding heat into two single strands. Next, cool down the mixture, and primer will attach to each single strand. Next, DNA polymerase will bind to the primer and start replicating. Then finally, two identical copies are formed. Inside polymerase chain reaction, in DNA sequencing and gel electrophoresis, there are no specialized machines required, but DNA can be analyzed more accurately by using them. Of these connections, we combine the biology and technology to find out more about genetics. Here's my bibliographies. Okay, that's the presentation of bioinformatics. Thank you for listening to my presentation.